Bismillahirrahmanirrahim. My name is Dr. Imran Wahid. Today I will discuss the few words about the structure of the kidney. We have already discussed it, and since we have already discussed it, I will take a brief review of what we have discussed before we start discussing the physiology of urine formation. So you can see in this diagram that we have a diagram of gross anatomy of the kidney. So this is the outer region which is known as a cortex. It is granular appearance because of the presence of cortical nephrons and glomeruli, and the inner portion is lighter one medulla. These are the lobules of the medulla, and these are the medullary pyramids, and they open into the calyces. So this is the renal artery which supplies blood to the kidney, and this is the renal from which the blood is drained out of the kidney. So this is the ureter. So these are a few words which are important before we start discussing the blood supply of the kidney. Renal artery supplies and renal vein drain the blood from the kidney. So it is very important to know that the gross structure of the kidney is designed in such a way that the arterial circulation can be accommodated to supply blood to the kidney. इसका स्ट्रक्चर इस तरह का बना दिया गया है कि कॉर्टेक्स जो है वो आउटर पोर्शन और ये मेडुला इनर पोर्शन दोनों को जो ब्लड सप्लाई है वो एडिक्वेटली जारी रहे. सो दिस फॉर दिस एडिक्वेट ब्लड सप्लाई टू द किडनी, nature has provided a renal artery and for a renal vein, but inside the kidney the structure is much more complicated. So it is very important to see, as I mentioned, that the cortex and medulla, they have different appearances on the gross anatomy of the kidney. Another diagram is very important. You can see here, this diagram actually shows how the blood is supplied to the kidney. What happened is that in the last figure we have seen that renal artery supplies blood to the kidney. So you can see here that a renal artery, it enters into the kidney and it divides into interlobar arteries. These are interlobar arteries. Interlobar arteries are actually present between the lobes I have shown in the previous diagram. So the interlobar arteries, they further divide into arcuate arteries. Arcuate arteries are known as arcuate because they form arcs like this. So these arcuate arteries then give rise to straight arteries, okay, and then straight arteries give rise to afferent arterioles. Renal artery divides into interlobar arteries, they divide into arcuate arteries give rise to straight arteries, and then straight arteries give rise to afferent arterioles. You are well familiar with the afferent arterioles. We have studied in previous lecture adequately about the afferent arterioles. So each afferent arteriole divides into a tuft or bunch of capillaries within the Bowman's capsule, which is known as glomerulus. Straight arteries are also called interlobular arteries. Okay, these are interlobar arteries, and straight arteries, these are known as interlobular arteries. Interlobular arteries. All the glomeruli within the cortex of the kidney, the capillaries of each glomerulus rejoin to form the efferent arteriole. You got my point, that this is the afferent arteriole. Afferent arteriole enters into the glomerular Bowman's capsule, a tuft of capillaries, and forms glomeruli, and all the glomeruli, they lie within the cortex of the kidney. The capillaries of each glomerulus rejoin to form efferent arterioles. Okay, so let us discuss it in further detail in a better diagram. You can see here, this is another detailed diagram. We have so far discussed that the renal artery divides into interlobar arteries, which divide into arcuate arteries. They give rise to straight arteries. Straight arteries are actually known as interlobular arteries, and from straight arteries we have afferent arterioles. Each afferent arteriole divides into a tuft of capillaries which stays close to the glomerulus to form glomerulus or glomerular capillaries. तो जो ग्लोमेरुलर कैपिलरीज होती हैं, दे लाई विद इन द कॉर्टेक्स ऑफ द किडनी. द कैपिलरीज ऑफ ईच ग्लोमेरुलस रीजॉइन टू फॉर्म एन efferent arteriole. So this is very important to understand that the route or the form taken by the blood after it passes through the efferent arterioles depends upon in which portion of the kidney the efferent artery is exiting, that is whether it is the cortical glomerulus or it is the juxtamedullary glomerulus. So here we can see that in those glomeruli which are present in the cortex, this diagram shows cortex and renal medulla, renal cortex and renal medulla. You can see that the renal arteries, as we have seen, that it divides into interlobar arteries and they drain through the interlobar veins. So here we can see that interlobar arteries, they divide into arcuate arteries. So this is an arcuate artery, this is an arcuate artery, this is arcuate vein. So arcuate arteries, this arcuate artery it basically gives rise to the straight arteries. Straight arteries are interlobar arteries. In cortical glomeruli, those glomeruli which are present in the cortex, we can see that from the straight arteries we have efferent arterioles. So this is the efferent arteriole. Efferent arteriole, actually efferent arteriole enters into the Bowman's capsule, form the tough capillaries which is known as glomerulus, and this is actually the course of efferent arterioles in the juxtamedullary glomeruli, not in the cortical glomeruli. Here we can see that in juxtamedullary glomeruli, the efferent arteriole exits and it tips down deeply into the medulla and then ascends upwards to join into the arcuate vein. These vessels, straight vessels deep into the medulla, are known as vada recta or straight vessels. They are basically formed by efferent arterioles of these juxtamedullary glomeruli. They branch to form these capillaries, and they are very important in concentration of the kidney. They form long loops, they run down into the medulla, surround the collecting ducts and run back to join the arcuate vein. In so far as cortical glomeruli is concerned, it is very simple. We can see that this is the arcuate artery and this is the efferent arteriole of the cortical glomerulus. It forms the same tough capillary, the glomerulus, and then from here efferent arteriole arises, which gives rise to a second network of capillaries which is confined to the cortex, and this network surrounds the proximal and distal convoluted tubules and cortical parts of collecting tubules. This is known as peritubular capillary network. Then it joins to the interlobar vein, which joins the arcuate veins. So it should be clear that in cortical glomeruli, the straight arteries give rise to the efferent arteriole form the glomerulus. From the glomerulus, efferent arteriole rise, which forms the peritubular capillary network, and they join ultimately to drain through the arcuate vein, interlobular vein and arcuate vein. But in juxtamedullary glomeruli, the efferent arteriole, after forming the glomerulus giving rise to the efferent arteriole dips down into the medulla in the form of straight vessels which run down and they ascend up, and they are straight parallel to each other, known as vada recta. They form long loops and then they run back and join the arcuate vein. So this is very important to understand this diagram. This diagram clearly indicates this is vada recta, this is arcuate vein, this is arcuate artery, and this is corticomedullary junction. So it is very clear that the fate of efferent arteriole is different in the cortical glomeruli and different in the juxtamedullary glomeruli. So to cut the long story short, very short, we can see that in cortical glomeruli, the efferent arteriole form the glomerulus and it divides into the efferent arteriole, which form the peritubular capillary network and then join the arcuate, interlobar vein and arcuate vein. In juxtaglomeruli glomeruli, the glomerulus gives rise to efferent arteriole, and then it dips down into medulla in the form of vasa recta, and then they ascend up and join the arcuate vein. So, to be very brief, we have read so far that blood supply of kidney is very important in the urine formation, because the tubules and the blood vessels run parallel to each other, are surrounding each other, so that tubules are in contact with the vessel in such a way that various substances, ions, solutions, waters can be absorbed from the bloodstream into the bloodstream, and they can be secreted or excreted into the lumen depending upon the requirement of the body. So, renal artery basically divides into arcuate arteries. Renal artery divides into interlobar arteries. They divide into arcuate arteries. Arcuate arteries give rise to straight arteries, which divide into interlobular arteries. Straight arteries are interlobular arteries. They give rise to afferent arterioles, which have different course in cortical glomeruli and different course in juxtamedullary glomeruli. In cortical glomeruli, afferent arteriole enters the glomerulus, form the Bowman's capsule, forming the glomerulus. It exits in the form of efferent arteriole and forms a peritubular capillary network which surrounds the proximal distal convoluted tubules and cortical portions of collecting tubules. Then they drain into the interlobar vein, and which joins the arcuate vein, finally draining into the renal vein. But in case of juxtamedullary glomeruli, you can see that the afferent arteriole forms similarly glomerulus, the Bowman's capsule. Then efferent arteriole dips down into the medulla in the form of vasa recta. Straight vessels run to each other. Then they run upwards, and they are branching. They form long loops and ultimately run back and join the arcuate vein. So, this is the importance of the blood supply of the kidney, that renal artery supplies and renal vein drains the blood from the kidney. So, we have discussed it. This is very important. So, we start next topic with this, that is the how urine formation starts. Urine formation actually starts in the glomerulus. This is the glomerulus. We have read it in detail. This is a simple diagram which shows that urine formation starts filtration of essentially protein free plasma through the glomerular capillaries into the Bowman space. 20% of the water and crystalloid constituents, solutes, molecules of small size of the plasma which enter the kidney via renal artery is filtered through the glomerular membrane. जो red blood cells हैं वो the red blood cells are larger in size, and other particles which cannot cross the glomerular basement membrane, they are not filtered. Otherwise 20% of the water and crystalloid constituents, solutes and molecules of small size of plasma which enter the kidney via renal artery is filtered through the glomerular basement membrane. Over 1 million nephrons are present in each kidney. So, we can see that in a simple diagram, it's a very good diagram which clearly shows how the substances are filtered from the glomerulus and under what changes they undergo before they are converted into urine. So, there are three processes. Number one is the process of filtration, glomerular filtration. Second is the process of tubular reabsorption, and third is the tubular secretion, and finally excretion in the form of urine. So, blood enters the nephron through afferent arteriole into glomerulus, which is a tuft of capillaries we have discussed, and this is the glomerulus, and this is the Bowman's capsule, and this is the tubule which has been shown in a straight manner. So, the high pressure within the glomerulus forces water and small solutes like ions, glucose, amino acids and waste products out of the capillaries of the Bowman's capsule, forming the glomerular filtrate. Glomerular filtrate, larger substances like blood cells, serum proteins are too large to cross the glomerular membrane, and they are not filtered and are retained in the blood. Hence, the initial glomerular filtrate is basically a diluted version of plasma. So, we should know that urine formation is starts at the end of the glomerulus, and the filtered fluid enters into the through the Bowman's capsule into the proximal convoluted tubule. Its convolutions are not shown here. It has been shown as a straight figure, straight diagram for the sake of understanding. So, we can see that 180 liters of fluid per day is filtered through the glomerulus into the Bowman space, into the proximal tubule, and as we know that the renal blood flow is at 125 ml per minute. The total plasma volume is about 3 liter. It gives us some assessment of how much plasma has to be filtered on daily basis. This is the proximal and distal convoluted tubule, and these are collecting ducts. So, filtration takes place in the glomerulus. So, this is the tubule and this is the bloodstream. You can see the reabsorption is the other process. In reabsorption, it is the process by which substances which are required by the body, they are reabsorbed from the tubular fluid into the surrounding peritubular capillaries and blood, and this is peritubular blood, which is shown here, this one. Third process is secretion. This is the process by which you can see the substances from the peritubular blood pass into the tubular fluid. Those substances which are needed by the body, they are reabsorbed in the proximal tubules, and those which need to be secreted in the out of the body, they are thrown or they are transported into the tubular fluid from the blood by the process of secretion. So, there are three major processes: filtration which take place at the level of glomerulus, reabsorption which take place at the level of proximal tubule, distal tubules, and secretion which take place at the level of distal tubules, convoluted tubules, and collecting ducts by other mechanisms. Excretion is when the substances pass out from the body in the form of urine. It is 1.8 liters per day. One important thing to note is that there are two routes for substances in the blood to be excreted in the urine. Either they have been filtered and not reabsorbed, फिल्टर हुए और रीएब्जॉर्ब नहीं हुए और उसी तरह यूरिन में आ गए, या फिर इट कैन बी सिक्रेटेड एंड नॉट रीएब्जॉर्ब, रीएब्जॉर्ब नहीं हुए लेकिन सिक्रीट हो गए किडनी फ्लूइड के अंदर और यूरिन में चले गए. इट कैन बी फिल्टर्ड. सो इन बोथ केसेस द सब्सटेंसेस विल देन बी एक्सक्रीटेड इन द यूरिन. ट्रांसपोर्ट मैकेनिज्म्स इन्वॉल्व्ड आर सेम as we have discussed: active transport, symport, counter transport, and involving the ATPase pumps, sodium potassium pumps. We will discuss it in detail. So, one thing is also important before we discuss the details of formation of urine and reabsorption and secretory processes in detail of these processes of reabsorption and secretion in tubules. It is important to discuss how the filtration takes place. Actually, the filtration pressure in the glomerulus is very important. So, the forces which are involved in filtration, they are very important. Forces which are involved in filtration of the blood through the glomerulus are very important. As I have mentioned you, this is afferent arteriole, this is efferent arteriole, this is the Bowman space or urinary space, and this is the glomerulus. So, according to the physiology, standard books of physiology, the pressure of the glomerular pressure is 60 millimeters of mercury. 60 millimeters of mercury is the glomerular pressure or hydrostatic pressure of blood in the glomerulus. Blood enters with this pressure 1200 ml per 12 ml per minute is the renal blood flow, as we mentioned. 540 ml of these are cells and 660 ml of plasma, water, colloids and crystalloids. Blood enters with 60 millimeters of mercury pressure, circulates in the glomerulus and leaves through the efferent arteriole. 1075 ml concentrated blood leaves per minute, 540 ml cells and 530 ml plasma, water and colloids and crystalloids. It is very important to know that blood is the hydrostatic pressure of blood is 60 millimeter of mercury, and the pressure, obviously the pressure which is trying to filter the blood out, there are other forces which are trying to keep the fluid inside the blood. So, the osmotic of pressure of plasma protein, which is 25 millimeter of mercury, is the opposing force, and second opposing force is the interstitial pressure in the Bowman space, that is the back pressure in the Bowman space. So 60 millimeter of mercury at the afferent arteriole end is the, you can see 60 millimeter of mercury is the hydrostatic pressure of blood, and this is the pressure which is filtration pressure, forces the blood out of the glomerulus into the urinary space of Bowman's capsule. But this 25 millimeter of mercury is the osmotic pressure of plasma protein, and this is the interstitial pressure which tends to keep the forces inside. So when the filtration pressure, we subtract these two opposing forces, so 20 millimeter of mercury is the main pressure, filtration pressure, effective filtration pressure through which the blood circulates in the glomerular and leads to the filtration of the blood in the form of fluid in the Bowman's space. So 20 millimeter of mercury at efferent, and this is the effective filtration pressure, but this value goes on decreasing when blood moves further. So we can see that here we can see that 125 ml filtrate or deproteinized plasma, that is without protein, per minute water crystalloid. There are no colloids or cells in it. They are filtered through the capillary and base and capsular membrane into the renal tubule. What is filtration barrier? Filtration barrier is the barrier which is responsible for filtration of blood from glomerular capillaries into the Bowman's or urinary space. This filtration barrier is formed by three layers. Number one, endothelial layer of the glomerular capillaries with large pores. Number two, there is a basement membrane, and third one is the layer of epithelial cells of the visceral layer of the Bowman's capsule, and these cells are called podocytes, which have large octopus-like extension or foot processes which are embedded in the basement membrane. This is the glomerular basement membrane, or known as filtration barrier. It does not require any energy of active transport, but the filtration pressure is simple method. It is a simple filter which allows the filtration process to proceed in a smooth fashion. So this is very important to understand this diagram. It shows the major forces: hydrostatic pressure of blood, osmotic pressure of plasma protein, interstitial pressure in the Bowman space, and we can see that blood is entering through 60 millimeter of mercury, circulating and leaving at this end. About 180 liters of dilute filtrates are formed this way every day. 180 लीटर के करीब 180 लीटर के करीब जो डाइल्यूट फ्लूइड है वो प्रोड्यूस होता है ग्लोमेरुलर फिल्ट्रेट के जरिए डेली बाय ग्लोमेरुलरी. सच फिल्ट्रेट कंटेन्स ग्लूकोज, salts, urea, uric acid, potassium, phosphate, sulfate, etc. in the same proportion as in blood plasma. It means the changes take place, but those items which are required by the body, electrolytes, sodium, glucose, they are preserved. 180 liters of dilute fluid. So the blood with this composition entering the glomerulus undergoes these changes and it is converted into urine. So this is very important to understand that filtration forces are very important. So with this we conclude our lecture, and in the next lecture I will discuss about the reabsorption which takes place in the tubular fluid and ultimately result in the formation of urine. So though if you have not listened my previous lecture, please listen the lecture to the end so that you can understand this what I have taught today. Please I request you to listen all the videos. You should subscribe this channel, click the bell icon so that you do not miss any video. So thank you very much.